Drug prices are rising. Can you imagine paying $50,000 a month for the medicine you need? That's how expensive some drugs are today. And it's complicated. There's a lot of reasons drugs are so expensive. And it's not all about the science. But one of the reasons is that they're very hard to develop and discover. The typical drug takes 10 years and costs about $2 billion to bring to market. So my research is interested in ways that we can speed up this process and develop better drugs more cheaply. So I use computational chemistry to study the molecules that drugs interact with. These are proteins. I first started thinking about this problem when I was in undergraduate organic chemistry. Now, some people love organic chemistry. Most people survive organic chemistry. And I was one of the survivors. But this was an important time for me because it's when I began to think about how drugs work at the molecular level. So caffeine, for example, which I'm showing up here. You drink a cup of coffee, caffeine goes into the body, moves through your bloodstream, and then it finds a protein in the brain and binds to that protein. And that's how you get the coffee buzz. That protein is called the adenosine receptor. All right, and basically all FDA-approved drugs work in this similar way. They interact with some protein, change the function, and that's how you get an effect. So I became interested in studying proteins. What are proteins? Proteins are encoded in our DNA. So the DNA has a 4-letter alphabet, A, C, T, and G. In the cell, these A, C, T's, and G's, they're translated to the protein language, which has 20 letters in its alphabet. These letters are amino acids. And when a protein is made, it's the arrangement of the amino acids that matter. So they have to be in a certain order, and this is called the protein sequence. So for example, a red to gray to blue sequence would be one protein, while a red, blue, gray sequence would be another protein. So each sequence leads to a different protein. All right, so the cell does this really easy. It translates the DNA from the A, C, T's, and G's to the amino acids that make up the protein. And when the protein is made, it's just an extended chain of amino acids. But to function, it has to fold to a certain three-dimensional structure. So you can see the cartoon shows a more compact version of the protein. Now it's just a cartoon, so I'll overlay it with a real protein. Right? And now I'll get rid of my cartoon, and we're looking at what a protein scientist would recognize as a protein. So we're able to look at this protein. By the way, this is the adenosine receptor, which caffeine interacts with. So caffeine would bind to the top of this protein, and that's how you get your coffee buzz. But we're able to look at this protein because the scientist was able to isolate it, purify it, get it to form crystals, and then bombard it with X-rays, and then interpret the diffraction pattern of the X-rays to put together a three-dimensional shape. So this and other protein structure determination methods are time-consuming, expensive, and they don't work for every protein. So if we understand the principles, though, of going from the protein sequence to the structure, we can bypass the experiment and run a simulation that gets us to the same place. And that's what I do in my research. So I work with software that was developed in the Dill lab here at Stony Brook that can take a protein sequence, we run a computer simulation, and then we get a protein structure. Now, this is great because sequences are easy to get. Structures are hard to get. So it's easy to get sequences. We develop an algorithm and try and predict protein structure. Here's an example of a protein where I was able to predict the structure using our algorithm. So this is a bacterial protein. Bacteria use this protein to make more proteins, and it's an example of what could be an antibiotic drug target. Remember, the proteins are important because we use them to develop and design new drugs. And here are 20 other proteins where I was able to predict the structure. So we know the reference, or the answer, and that's in gray. And then we have our prediction in blue, and we match them up and see how well they match. Now, I'm showing a lot of successes here, and this is an exciting result, but there's still a lot of issues. So I'm trying to make the algorithm better, more reliable, so we can deliver structures so that people can develop drugs and eventually lower the cost of drugs. And that's my vision for better living through chemistry. Thanks.